Oil and water refuse to mix. In a watery environment, lipids immediately aggregate into isolated, insoluble blobs. This physical reality is a significant hurdle for human biology because the entire gastrointestinal tract is a highly aqueous environment. Dietary lipids, triacylglycerols, cholesterol, and phospholipids all share this characteristic. Their structures contain long, hydrophobic regions that aggressively repel water. A standard diet introduces 20 to 30 grams of these lipids daily, while a typical Western diet can contain 2 to 3 times that amount. Without biological intervention, these 30-plus grams of fat would remain as large, unabsorbable clumps that the body's enzymes could not access. To process these molecules, the body must employ a specific sequence of mechanical and biochemical tools to force lipids into a water-soluble state for transport. Lipid digestion is a process of overcoming surface tension to allow for enzymatic cleavage and eventual absorption. The process begins in the mouth and continues as the food enters the stomach. Here, the body introduces the first enzymes, lingual lipase from the mouth and gastric lipase from the stomach. These lipases are acid-stable. Unlike many other enzymes, they remain active in the low pH environment of the stomach, which typically ranges from 2.5 to 5.4. This table identifies these as the first line of chemical attack, categorized by their ability to function in acidic environments. Their preferred substrates are triglycerides, with short and medium-chain fatty acids, which they break down into free fatty acids and diacylglycerols. This stomach-based digestion is particularly important for newborns, as milk and butter are rich in these shorter-chain triglycerides. While up to 30% of triglyceride digestion occurs here, larger long-chain lipids require a neutral environment and more specialized tools to be fully dismantled. When chyme moves into the duodenum, bile enters at a pH of 7.7. This neutralizes the stomach acid, creating the proper environment for pancreatic enzymes to function. Before those enzymes can work, the large fat aggregates must be dispersed into a fine emulsion. Initially, the fat exists as large aggregates with a very low surface area to volume ratio, as seen in the top of this diagram. To resolve this, the body introduces bile salts, such as sodium glycocholate, which act as biological detergents to lower surface tension. Bile salts are amphipathic. Their hydrophobic portions intercalate into the fat, while their hydrophilic domains remain at the surface, facing the water. This mechanical action breaks the large aggregates into smaller droplets, forming an emulsion where the fat is shielded by a water-soluble coating of bile salts. This emulsification significantly increases the surface area available for the chemical digestion that follows. With the fat now emulsified, pancreatic lipase and its colipase can finally access the triglycerides. Pancreatic lipase hydrolyzes the fatty acids attached to the first and third carbon atoms of the glycerol backbone. This leaves behind 2 long-chain free fatty acids and 1 molecule of 2-monoacylglycerol. To move these insoluble products through the gut lumen, the body incorporates them into a mixed micelle. Its exterior architecture is formed by amphipathic bile salts facing the water. The hydrophobic cargo, fatty acids and cholesterol, is packed into the interior core. The mixed micelle serves as a temporary water-soluble vehicle that delivers these insoluble lipids directly to the intestinal wall for absorption. Upon reaching the mucosal cell, the micelle releases its cargo, allowing fatty acids and monoglycerides to pass passively across the cell membrane. This diagram tracks the entry of these digestion products across the brush border of the intestinal mucosal cell. Once inside, the cell reassembles these fragments back into triacylglycerols through a process of reesterification. To transport these rebuilt fats through the blood, they are packaged into large lipoproteins called chylomicrons. Chylomicrons are formed by coating the triglycerides with cholesterol esters, phospholipids, and specific apoproteins, such as B48. Because chylomicrons are too large for capillaries, they are exocytosed into the lymphatic vessels, or lacteals, eventually entering the systemic blood through the thoracic duct. However, short and medium-chain fatty acids bypass this entire lymphatic route, as highlighted here. They diffuse directly into the portal circulation. Their shorter chain length makes them soluble enough to travel without micelles or chylomicrons, providing a direct shortcut to the liver. In this system, the carbon chain length of a lipid determines the specific metabolic routing the body must use to transport it. This summary diagram traces the full path from the intestines to systemic tissues like muscle and adipose, where fat is used for energy or storage. Following a high-fat meal, the literal presence of chylomicrons in circulation can give blood serum a milky appearance, a condition known as postprandial lipemia. These standard clinical values reflect the balance of lipids being transported through the blood at any given time. For the liver to properly mobilize these fats and prevent them from accumulating, it requires specific lipotropic factors. Factors like choline, methionine, and omega-3 fatty acids are required for normal fat mobilization from the liver. A deficiency in these factors can lead to fatty liver disease, as the liver becomes unable to export its processed lipids. Once fats reach the tissues and are broken down into acetyl-CoA for energy, they enter the TCA cycle, but only if carbohydrates are available. This leads to the metabolic principle that fat is burned under the fire of carbohydrates. If carbohydrates are missing, as in starvation or diabetes, the acetyl-CoA is instead diverted to form ketone bodies, like acetoacetate in the liver mitochondria. Lipid metabolism is a highly coordinated sequence of mechanical emulsification and molecular packaging that ultimately relies on carbohydrate availability to reach its final metabolic destination.